okay? Water, solutes is stuff dissolved in the solvent. And again, the amount you have in there is a concentration gradient. And with cells, again, with the membrane, we've got concentration gradient on either side. Okay? And again, that plays a role, of course, in what we're going to talk about now about the transport. Does everybody understand the environment and all that? And you can, by the way, say no. Can't say no.
Membrane transport?
Say it again?
Are we on membrane transport on like
Yes. Yes. All right, so let's talk about passive transport, then we're going to talk about active for a second. All right, so passive transport, which is also called diffusion, is the movement of substances from high concentration to low concentration. So just like we're talking about over there with sodium and with potassium. Okay? Now, the thing about high concentration and low concentration right here and the movement is it does not require cell energy. It does not require ATP for this to happen. But we do have energy involved, and this energy is kinetic energy. This is the kinetic energy of the molecules themselves. And so basically this kinetic energy refers to the fact that if you take a population of sodium ions and you have them sitting in the solution, they're not sitting static still. They're moving. They're always moving. Everything is always moving, right? And as they move, they begin to hit each other and bounce away. They begin to hit each other and bounce away. And the more you have in one place, the more often they're going to hit each other and bounce away. And the tendency will be for them to move from where there's a whole bunch of them bouncing off of each other to finally kind of moving off to where there are few of them. And again, that's energy, but it's not cell energy. So that picture on the left just illustrates with the little arrows, the little balls aren't just sitting. They're all like kind of what? Buzzing around. They can't sit still. Can't sit still, right? So as they're kind of moving around randomly relative to each other, sometimes they're hitting together and then they bounce away. They bounce off of stuff, bounce back, and there's just all these collisions going on. Now, when you think about these moving in the context of the cell, when we have a membrane right here, you've got a certain number of molecules on this side, you've got a certain number of molecules on this side. All of them are bouncing off of each other. But as these bounce off of each other right here, they kind of get ricocheted, and some of them make their way to the other side. As these are bouncing around over here, they kind of ricochet, some make them back this direction. And the thing is if you have a whole bunch of them right here, but fewer right here, you're going to have more collisions on this side. And as they collide, they're going to start to drift over here to the other side, along the concentration gradient. You still got collisions over here, but if there's fewer over here, even though some get directed this way, most of the movement is going to be that direction. And it will achieve an equilibrium. Point is that there is energy, but it's the inherent movement of the cell energy, just sort of what? Say buzzing, right? They're just jiggling. And as they do so, they're bouncing off of each other. No ATP. That's only involved in active form of transport. Okay? All right, so this is just illustrating that. Same thing. You've got all these orange balls on this side, nothing over here. Here's the membrane. They're just bouncing off of each other. They start to diffuse across. And then pretty soon they do reach equilibrium. And over here, again, we got them going both ways, but for every one that goes this direction, we have one going this way here. Initially, most of the collisions are here, so most of the movement going that way. This is to illustrate that solutes are individual from each other, independent. So the orange ones reach their own kind of equilibrium through collision, has no bearing on having purple ones there. Purple ones reach their own equilibrium independently of the orange ones. They don't influence each other in terms of their movement, okay? Same thing if you put dye in water. You put the red dye in the water, the dye molecule bounce off each other. Pretty soon, of course, you're gonna have a completely, colored water. If you put two colors in there, even though they won't remain two colors at the end, those two colored molecules will again move independently to reach their own equilibrium, okay? Because again, the red one is not influenced by the presence of the yellow is not influenced by the presence of the red. How about Cookie Monsters? So cookies come out of the oven, it's high concentration right there at the oven. And then of course it just sort of diffuses in the air throughout the house. Right? Here we come. Yeah, here we come. Anyway, so again, diffusion in air, same thing. That's why they put odorant in gas. You know, your gas, so that when you've got a gas leak, you can tell. Right? Because otherwise it's odorless. All right, so anyway, that's the idea of diffusion. And again, the molecules just moving, you know, to a lower concentration area just simply because of their inherent amount of movement. Right? All right, so anyway, a couple examples here for diffusion. All right, so simple diffusion, first of all, is basically moving the molecules through the phospholipid membrane. So you can see we're not really showing any protein on this one, so this phospholipid bilayer is what we're talking about. So that means that these molecules are ultra small, but they're also lipid soluble. That means they can get into the membrane with no trouble. So oxygen and CO2 up there, our two respiratory gases, are a good example of molecules that can actually just go through the plane of the membrane. So simple means it needs no assist, there's no protein, nothing like that, in order to move across the membrane. Okay? Also, let me point out that, you know, fat-soluble vitamins, even though vitamins themselves tend to be fairly large structurally, if they are lipid-soluble, still they come through the membrane. Cholesterol itself, not showing right there. It's a lipid, so it would just kind of dissolve and move through the membrane. Okay, that's simple. Phospholipid straight up. All right, facilitated diffusion means that you've got a protein. And so, like for the sodium and the potassium over there, you have a channel. This is what allows sodium to move from the outside to the inside, or the potassium to move from the inside to the outside. Because without that protein, sodium and potassium being charged can't move through the phospholipid. So this is facilitated diffusion. Still along the gradient, okay? High concentration to low for sodium, or from the inside out for potassium. We also have a little bit bigger version of facilitated diffusion, and this is kind of how glucose moves into the cell. And so we have this protein transporter right here that you can see is really an integral protein that can actually change shape. And so we've got high concentration of glucose on the outside of the cell and low on the inside. And this protein will allow glucose to fit into it. And when it does so, the deal flips from this direction to this direction, releases the glucose into the cell, and then it flips back the other way to pick up another glucose. So this is facilitated because glucose, first of all, too big, not lipid soluble, and otherwise not ever going to fit through this part of the membrane. But this is passive because we're going from high concentration here to low. But if it involves a protein, it's facilitated. There's an assist with it to move across the membrane. And so both this and this right here is also facilitated because they can't move without the protein. All right, and then the next one is the one that is the most tricky, osmosis. So this is going to be the movement of water across the membrane.